Thank you for a nice introduction. I think you're very tired and very sleepy and very hungry too, right? So I have a 25 minutes and I'll definitely finish it before 6 o'clock, okay? So please wake up and then focusing on and because it's the last presentation. So once I sent to my title of the presentation, I'm trying to collect data, and then I communicate to many scientists in Korea. So I get rid of the biofertilizer because we have too many biofertilizer companies and products, and then so I get rid of this one, and then I only focus on bioprotectant, like a biocontrol agent. Then I'm also asking myself two very fundamental questions. Why I'm working on plant microinteractions? What's the final goals? I think it's the simple thing is make plant happy. Actually, I don't know how the plant have a feeling like this, but I'm trying to figure out what I'm going to do. That quote means plant is not unhappy, sometimes, not always. So why is it unhappy? Like human beings, plant also under stresses. So what kind of stresses? Is biotic and abiotic stresses. So as you can see here, as plant got a lot of the biotic stresses like virus, bacteria, and fungi, insects. So then human beings try to control those kind of the biological factors. How? This is in the textbook, there are several ways to control the plant pathogens and insects. There are cultural control and chemical and biological controls. So today I'm going to just focus on biological controls. There are 4 mechanisms, including antibiotics and hypoparasite and competition and induced resistance. So luckily, Pam already mentioned about the whole introductory things in how it's going on in biological controls. So I may skip almost everything. I save a lot of time, so it's finish it early a little bit. So this, I think it's just focusing on the consumers really required to really clean and low residues and also reduce the environmental factors. Okay, even also improve the worker safety too. So the gradually increase the interest of the many products, and also the almost a 10% increasing rate every year. I skip this one. Actually, in Korea, we have the environmentally friendly farms is 4.9%. It's very teeny bitty percentage, but still there's an increase because the consumer really require this kind of product. So I hope it's increasing gradually. In Korea, in pesticide, mostly focus on the apples and pepper and cabbage in citrus, because the kimchi is a big product for us, and we are working on those Chinese cabbage and pepper things. And also, this is anthracnose, bacterial disease, and oomycetes and rhizoctonase target pathogens. I will talking about a little bit about the bacterial diseases later. Let me introduce a little bit about a little bit difference from other countries in Korea. We have very unique regulations. Actually, we do have 3 different products if you want to selling some product, microbial product. The first one is like biopesticide, like a biocontrol agent. It normally takes 3 years. It spent 300 to 430,000 US dollars. It's a lot of money and a long days. And if you want to selling biofertilizer, this is only 6 months and 5,700 US dollars. Actually, there is another category to organic agro materials for disease controls. This is a little bit lower level of biocontrol agent because this spent 6 months and 21,000 US dollars. So most of the companies interested in these categories first, if it's really effective, and move to biocontrol agents. So it's kind of intermediate product you can selling in Korea. So there is mostly 10 big companies. I'm sorry, in Koreans. We have the Syngenta and Bayer, and also there's a Palm, Hannong's, and then Gyeongnong's is the biggest one, I think. So I'm not going in detail. I'm just point out here, biopesticide market is 1.2 million US dollars. It's not too big. Among them, there's a 55 is imported biopesticide. So it's only 45% is our own markets. So I'm just interested a little bit about what I'm going to do in my laboratory because it's I'm very interested in plant growth promoting rhizobacteria, so-called in the PCPRs. This is introduced by Joe Kraft, who is my advisor, in 1986. This is just isolated is a good bacteria from the root rhizosphere around the root in the plants and introduced to plant growth promotion and biocontrols. So while we're interested in this bacteria, because once we apply it in the field, we can see really good protection of the plants compared to controls. This is caused by some diseases or insects. So now also we can also increase yield. In my PhD study, I found one of interesting phenomena because you can see, look at carefully here. This is normal petri dish. There's one barrier here. So we inoculate the bacteria one side, and the other side is we inoculate in plants. So we can cover it in the reed and then tightly sealing with this one. And 2 weeks later, we can see this kind of phenomenon. So one bacteria cannot directly interact with the plant. But you can see here, there's a water treatment that looks like this, and bacteria treatment that looks quite bigger. One explanation is by plant volatile release from one side to the other is increased plant growth like this. So bacteria smells increase plant growth. So what kind of smell? Is this a lot of the bacterial volatiles characterized is, I can introduce just one. Trisylbutanediol is one of the high amount of the bacteria volatile released from these bacteria. So we mutated the bacteria is loss of function of the plant growth, promotion, and also induce resistance. If it's increase of volatile production, is quite better than wild types. So we also applied this volatile compound in the field. We can see good protection against virus diseases. Another interesting point, that there is a different stereotype of this volatiles has different effectivities. We conducted several years in the field conditions. In biological controls, we have the fundamental problems because mostly we are searching like this: reductionism-oriented biological control agent screening. Like we isolate the bacteria, then we screened in the greenhouse and the field. So sometimes it's working very well in greenhouse, it's not working in field conditions. Why? In 1983, it's really long time ago, there is a book in the Nature and Practice of the Biological Control of the Plant Pathology. Is written by Cook and Bakers. And one page shows like this: more than 100 years after introduction of the soil disinfestation, this kind of biological control, more than 50 years after Stanford classic publication biological control, we are frustrated by large gap between promising result in greenhouse but fair in the field. It was 1983. I mean, still we have the same problem here. So why these things still have problems? I think many people already, mentioned about this one. Because we understand the cultural bacteria, but we do not understand unculturable, or once we introduce bacteria, what happens in the real conditions. That is the problems. So I think we can consider new technologies like genome sequencing or synthetic biologies and metagenomics. And we have to understand the real condition in the field, I think. This is one of the examples of what we did in my laboratories. So one day we visited in tomato farmers in totally different areas. We can see very interesting phenomena like we see this totally wilted tomatoes. This is caused by Rhizostomus solanacearum, which is bacterial wilt. But very interestingly, right beside the plant is very healthy. Classical the philosophy in plant pathology, it should be also showing same symptom, but it's very clear in right beside plant. It also same thing happens in Mississippi in United States too. So why these things happens? We try to isolate the pathogen population in diseases plant and healthy plant. We can see exactly the same number of the bacterial pathogenic bacteria number in here. So that means the pathogen population is the same, but one is diseases, one is healthy. What's the difference? We isolate some bacteria and metagenomic analysis. We figure out some bacterial composition is different in health and diseases. So simply say, this is this biosis was occur. This is a symbiosis, eubiosis. So that means good bacterial composition make plant healthier. So we try to isolate some bacteria as compared to metagenomic analysis. We found the 4 different bacteria is very critical. So we tested in individual and 2-3 combinations. So the good thing is the only 4 combination is a very strong control phenomenon. So that means the minimum bacterial composition is very important when we apply it in the field conditions. So we can check the genes expression. We also interested in how can we apply this bacterial composition in the other aspect, like Pam already introduced. We also interesting on how methane reduction introduced by some microbiomes. We already have the data, like we are interested in promote growth of a methane consumer. So methane consumer means methanotropes. The problem was methane, methanocene is difficult to grow in large-scale productions. So we cannot apply in the rice field. So we change our concept. We just try to isolate helper bacteria of methane consumers. So we isolate any heterotrophs like Burkholderia bacillus. Some certain bacillus and Burkholderia species is significantly help methane consumers' growth. So we can successfully reduce methane reduction even in the field. So in microbiome is really great potential for future agricultures, I think. Let me briefly introduce in what kind of the crop protectant, which is a biocontrol agent in Korea product. In early 70s, 80s, we just stages for selecting antagonists against the target pathogens, and then adapt new information and technology from the abroad. And 1990s, just kind of developing phases. This is we are interested in plant growth promoting rhizobacteria fungi-related studies, and also introduce induced system resistance and apply the large-scale production of the microbes. And 2000 is a mature stages. We formulated the expanding markets and mechanism studies through the multi-omics technologies. And after, 21st centuries, we are working on microbiome, how can we apply it? But still, we do not have really a solid evidence and product for microbiome related mechanisms. In Korea, recently, we are really interested in this disease. So because the bacterial disease is a really great market, because this is a fire of light. This was introduced in 2019. This is introduced at the beginning of right below in Seoul area. This is really spread a lot. It's a big impact in pear and apple production in Korea. So we just apply in chemical antibiotics, but it's not working very well actually. Keeps spreading even in today's. So we try to bacteriophages and some bacteria bacillus, even in selenite, who the Pam already mentioned about that. It works well, but still it's not perfect protections. We have still problems. We also do have the different products like Nevobacter also have some effect in this the pathogens. So we also another problem is pine wilt diseases, because as Korean really like the pine trees, and we can have a lot of the product for the build the house and some others. But the pine wilt disease is caused by the nematodes and also transmitted by some insect beetles. So this is a really big problems. Or we have the ivermectin and sulbexins, but we still have a lot of the other products developed right now. The chemical control is not working very well. So we also have the BT, we already mentioned a lot. We also have another BT product like this. There is another product I can just introduce this is we call the Choong-Che sak. I don't know in English. So this is one of the interesting target because this is a Buveria persona. It's very common for insecticide fungi, but this is only target in soil the insect. So trip is in the life cycle there's only stay in the some pupa stage in the early larva stage in the soils. Only target for these guys. So it's successfully controlled these turnips. There is also many above ground pathogens in here. So there is another one is Bacillus valenciensis EXTN1. This is developed by the RDA here and with collaborate with Dongbu Hannong. This is target for solvent pathogens and mechanisms that induce resistance. They're also selling with some biofertilizer too. So we also interesting the ginseng productions. This is a target for Cylindro simplicum. The product acne is also working well. We also target in many nematode problems. We have the Bacillus subtilis and also Monacosurium product. This is also working well in the outside. We also have some fungi production like product like Trichoderma is also working well. The recently also release is Mesona is one of the interesting for me because it is against the abiotic stresses. This is a high, low temperature and water and salt accumulations. Okay. I think this is the last slide. Still, requirement for future plant protectants and consistent and durable effect and overcoming stability of the previous bio product and plant protectant portfolio is really important. Sometimes one product control almost everything. It's not good strategies because it should be in specific targets, and then we can have the portfolios. Preparing new emerging diseases caused by climate change too. Okay, let me also acknowledgement is many people provide really valuable information like Young-Yun Jung and Young-Chul Kim and Mi-Kyung Sang and Jin-Chul Kim in here and Taek Soo Sin. Thank you so much.